Hello, this is Mr. Young, Ecotech Lab. I have today Ms. Ramirez from UPSM High School. She's going to lead us through a session on extraction of DNA from strawberries and bananas. Okay, this is going to be interactive, y'all. So make sure that if you have questions, just stop us as we go forward. But this is going to have some baseline information too about DNA. So what I'm going to do now is I'm going to share a screen with you, and you tell me when you see it. Tell me when you see it. Do y'all see
I
it
see
yet?
it.
Everybody
I
good?
see it.
Okay. All right, then. So what we're going to do is we're going to have Ms. Ramirez kick this off, and then I'll come back and chime in. Okay, Ms. Ramirez is here.
Good morning again. So I'm gonna start off with the DNA extraction from strawberries. So extraction, when you think about the word extraction, you think about what? What do you think about, Brianna?
Taking something out.
Joshua, you said extract something from the strawberries. How can I pull out DNA from a strawberry? So that's a question. That's a big question that you need to ask yourself. How can that happen? So I'm going to go over a real short slideshow to give you some basic information about that. All right, so you know DNA is found in every living organism. And fruit is a living what? It's also called what? A producer, right? So it's part of living things. So you know DNA is also living. And so what we're going to do, we're going to isolate and extract and isolate by using this simple technique. Something similar to what you said, Brianna. I'm going to use mortar jar, some test tubes, some alcohol, fresh banana, fresh strawberries, and water, and go from there. All right, so after the extraction, after DNA has been extracted, you're going to be able to see this with the naked eye. So we already know that cells, the DNA is represented in cells of all living organisms. And also DNA can be extracted from human cells or it's for testing, for genetic testing in laboratories. So they use it for genetic testing, they can use it for forensic science, they can use it for so many different things to actually obtain the DNA. So scientists always wanted to separate unwanted substances from the cell. So that's what I'm gonna do here. I'm going to separate the unwanted. I'm gonna break down some tissue, break down the wall, break down, get to the cytoplasm, and separate the stuff that we do not need so you can actually see DNA from the naked eye. So we have Mr. Young that's going to give you a few facts about DNA.
Okay, all right. Okay, guys. So, the game plan on this is that as we go back and we look at the DNA, we know that it has a actual structure. We're going to talk about this DNA structure real quick. Do you notice a couple things? What's the first thing that you notice, Josh, with the DNA?
ATCG.
Right. Okay. You jump. Yeah. You okay? You talk about the 4 base pairs. Absolutely. What else do you notice, Brianna? You notice anything? I built this thing, by the way. We built it from scratch on this one.
It spirals upward, like it twists upward when it goes around. So
bingo. So you got this. You got this spiral like a ladder, almost like a ladder. So if I start looking at those base pairs that Josh talked about, guanine, cytosine, thymine, adenine. Those are 4 base pairs. So back to what you just said, Josh. Let's say A to T, C to G. Say that again, Josh.
A, T, C, and G.
Right. And what does that tell us? What does that tell us about that combination? Are those the guys that are brought together or are they separate?
Those are the A and T are what are combined, and C and G are what combined, but those two are separate pairs.
There it is. There it is. So you guys look at that little red dot here. You see this connection here? Deoxyribose. Deoxyribose. What about the purple piece? What do you think this is? What do y'all think this is? Phosphate. It's phosphate, right? So deoxyribose phosphate, you have this long chain. So Watson and Crick really designed, or at least got credit for this work in terms of what the DNA model looks like from an infrastructural standpoint. The game plan is when we go to extract from the strawberry and the banana, this is the structure that we're actually taking out. How many chromosomes does a human have? Should be 46, right? 23 from mom, 23 from dad, right?
Yes.
Yeah.
So Ms. Ramirez is gonna get started, looks like with strawberries first. I am going to step to the side and I'll be back. I'll help out where I can, okay?
All right. First thing, I'm going to start off with my strawberries. And I am going to take 50 milliliters of water. And I'm going to crush them, starting kind of slowly. You want to break that cell wall. This will take you about a good 2 or maybe 3 minutes, depending on how hard the strawberries are, how quick you can get it broken down. So I use 4 strawberries, and again, 50 milliliters of water. How would you do this at home if you had a chance to do it at home? You don't have a mortar and pellet. How would you do this at home?
You could use a spoon and a bowl.
My next step. So I have a beaker, I have a funnel, I have some cheesecloth, and I'm going to pour this over here to see how much DNA I can get from the strawberry. I'm just gonna put it back.
Can y'all see that, how it's filtering through?
Yeah, can I add something?
Mm-hmm.
I just want to say that the liquid at the bottom is the DNA and other cellular substances for you to collect.
Right.
So the DNA is seeping through the cloth?
Yes, it is. Push it down just a tad bit. Get a few more drops on there, and we should be all set. All right, so now we're gonna transfer this in a test tube, because after I use my surfactant, you'll be able to see the DNA actually separates. Next, I'm gonna use this extraction liquid, just a few drops.
What's inside of it that allows it to extract?
Sodium chloride, water, and dish detergent to kind of break down, separate. Dish detergent kind of separates the cells. So next, I'm going to use 1 milliliter of alcohol, preferably 70% of alcohol, using a pipette. Try to get all the air out. All right, let that sit for about 2 to 3 minutes. Let's see if we capture some DNA off the strawberries. I'm gonna use my stirring rod to see if I can pull the DNA off. See as it starts to separate. Wow, this one starts to separate already.
Is the alcohol sitting on top of the solution?
Yes.
The alcohol is what separates the other cellular substances from the DNA. So the DNA will be floating on top.
Mm-hmm.
Do you think it makes a difference to add the extraction solution as opposed to adding them individually?
I think it should be combined. If that's what you're asking me, instead of individually, yes, to speed up the reaction.
Y'all can see how that DNA is being separated there, right? So the first thing we want to say is, well, can we do this with a banana, right? Brianna, did you ever come back with that data on the strawberry and the length of the DNA strand?
I didn't get the length, but I did get the chromosome. It was like
What's the
it said it
chromosome
had
count
8
on
copies.
this? Huh?
It said it had 8 copies of chromosome. I didn't get the length though. I looked
Okay.
and looked and I ain't find it.
Okay, well, that's fine. So 8 chromosomes for a strawberry? Write this down, Victoria. How do strawberries reproduce? Put that down in your question from this experiment, because typically we want to know if it's a living organism, then there's a reproduction process.